All right, where did that go? Perfect. All right, so let's get into the rest of that going on here. So just a little bit of review from last time. So the Booth paper, some really big points about it, and I'm going to be going into just a little bit more thrifty gene theory, and really his whole model of like physical inactivity I think is super important for us to know. So the basic framework for gene regulation was selected for during an era of obligatory, meaning you don't have a choice, of physical activity. Does anybody remember how many miles I said the average hunter-gatherer female ran or walked, and the average male hunter-gatherer ran or walked? Yeah. Yes, so like 7, 7.5, something like that for females, 9, 9.5, something like that for males. It's like, shoot, on Saturday, I walked my dog 7 miles, so I'm almost a Paleolithic lady. I'm working on being better with that. And then the next day, I only did 6, so that was kind of lame, but whatever. I got tired. Here, sedentary, being sedentary, equals death in like the general environment that we evolved in or that we were thrust into, right? Enjoyment of activity was essentially inconsequential. And on average, it seems like only 5% of the population even really enjoys or does the requisite amount of physical activity. And most individuals actually end up not doing the requisite amount, meaning like 150 minutes of moderate physical activity or walking 3 miles per hour every week. And I can test you on that. That's another class where I test that mostly. Next thing, humans aren't fragile or resilient. We actually need to be stressed in order to maintain normal physiology. That's really what all of this is about. Probably the most philosophical stuff I'm going to say in this class. And like here, let's move on just a little bit. In the like paper by Frank Booth, they talked about the thrifty gene hypothesis or thrifty gene theory. I forget what nomenclature they actually use there. I think it's hypothesis, but I don't really recall. So let's go through this really quick. There are essentially feast-famine cycles, meaning that like there was a long period of time where we weren't eating and we were chasing down like buffalo or mammoth or even other humans. That actually did happen, like a little bit at least. And then we would have a massive feast. So here, let's look at this. So feast right here, intake of glucose and fat. Now a lot of what actually happened in the environment, we would actually convert protein to glucose quite frequently. That's a process called gluconeogenesis. I'm going to tell you much more about that in bioenergetics, but right now I just want you to be aware of that. Because really, like, access to different like carbohydrates, which that's what glucose is, was more of a seasoning, but almost all year you could get fat and protein. So eat a massive amount of that, and then whenever you eat a massive amount of that, something interesting would happen. Right here, insulin resistance. Does anybody know what insulin resistance is? So whenever I say it, I want everyone to think type 2 diabetes. Is everyone good with this? So here's like a radical idea. Insulin resistance is probably bad for you. So insulin is a hormone secreted by the beta cells of the pancreas that is essentially the key to like the lock on a muscle or fat cell to get glucose and fat and even amino acids inside of the cell. Now, whenever we start getting insulin resistant, say the muscle doesn't respond to that insulin anymore. It's like, hey, we're full. We don't need any more glucose or amino acids or fat or anything like that. And then that ends up being a problem. That's why people who are diabetic, their circulating like plasma glucose starts elevating, going up. And that's really bad for like a myriad of things. So I don't have time to talk about it, but like free radical damage is kind of a big thing with it. And this is actually, or at least seems to be, a positive evolutionary adaptation, because we would be starting to store all of this glucose and fat inside of a muscle, and then once the muscle is full, we adapt, start becoming insulin resistant, and we start storing it in fat cells. And converting everything to fat and storing everything in fat cells, what is that good for? Energy, yes, yes. So like really basically the whole reason why we store fat is for long periods of time where we don't have access to food. Does that make sense to everybody? Right? So if you compare us to like other like primates or hominids or anything like that, we are by far the highest in percentage of body fat. And like a chimpanzee, has anyone ever seen like a chimpanzee with like mange? Meaning like all of their fur comes off? Like, I'm not going to show you a picture of it because like you start seeing like reproductive parts I don't really want to show in front of a whole bunch of people, but they are ripped. So are gorillas, so are all of them, but all of us, we tend to be like 10, 20, 30% body fat or higher, whereas like chimpanzees are 5% or less. And that makes it to where we can go a much longer period of time without eating. Is everyone good with this? Right? So kind of a radical idea that I'm saying right here. Throughout evolutionary history, insulin resistance is a positive, bless you, adaptation. So we funnel all of the energy towards fat cells so that whenever we're starving, we have fat cells, or we have more fat to pull out. Is everyone good with that? Good? Okay, so that would happen under the feast condition. So effectively, there's like these thrifty genes, so-called thrifty genes, that make us better at holding on to fat and like less good at burning it and all of those different types of things. So essentially, people who are the best at storing body fat survived. Because whenever a famine hit, all of our lean ancestors didn't make it. Right? Does that make sense? Cool. So that's, and like there's some like, I'm not going to talk about environmental differences. Then here at the end, after a long period of essentially not eating, like there would be famine and physical activity, meaning muscle contractions or just not having a lot of energy around, that would make it to where our skeletal muscle and different tissues are actually more sensitive to energy. So this right here, like GLUT4, I might write that down. That's the primary way how we get glucose or carbohydrate into a muscle cell. And like I'll talk more about like how that whole thing is regulated. But just right now, since so much of this exercise physiology stuff and biochemistry is essentially a different language, that's why I'm trying to use it as much as I can right now. Because GLUT4 and I don't assume anybody really knows what these mean, but that's kind of where the whole field of exercise physiology is going, measuring these different like proteins and transporters and stuff. So everyone good with the thrifty gene things? I mean, I'm good with it. I understand it, but hopefully you do. What's up, Sydney? It is a transporter inside of a muscle cell that is stimulated by not eating or physical activity. And it's essentially like a shuttle to get glucose inside of a muscle cell. We have other ones like GLUT1, GLUT2, GLUT3, and like they're expressed in different tissues like the central nervous system or whatever. But GLUT4, that's the super awesome one for muscle. That's how we get jacked. So here, just a couple of things that I wanted to talk about. Like this one quote, I took it from the paper because I really like it. And if you don't know what this term means, I'm going to explain it to you. Modern humans are characterized by a striking sarcopenia, meaning that we're so sedentary that we are essentially deficient in quantity of muscle mass on the body. So in terms of proportions on how much muscle mass a hunter-gatherer had versus how much we have, typically, unless you're like one of these yoked-up bodybuilders, they have a lot more because they're using it all the time. That's pretty cool. They also had a greater capacity to oxidize fuels. So that is called mitochondrial density. So write down mitochondrial density. I want you to know that. All that means is the amount of mitochondria inside of a muscle, or inside of a muscle cell, technically. And here I'm going to show you a proxy for that as this goes on. It helps the cardiovascular system. They talked about this a fair bit. For example, hunter-gatherers don't get heart disease. So that's a kind of cool thing. Like they're eating grass and red meat as they possibly can, and they're essentially sprinting and throwing spears all the time, and they never get heart disease. Now, here, I'm going to talk about like a bit of a myth really quick. What's the average age that a hunter-gatherer would get to? Does anybody know? What's up, Nick? Yeah, somewhere like mid-30s to like mid-40s, somewhere about there. So does anybody want to like die in their mid-30s or mid-40s? No? Okay, well, that makes one of us all right. So there's a problem in counting this because they had super high infant mortality. So does anybody know how like averages work? Right? So you add up the lifespan of everybody and then you divide it by the amount of people in it. So if I'm adding up the lifespan and I'm adding a bunch of zeros because people died in childbirth, that's going to drastically reduce the mean or the average life expectancy. Is everyone good with that? In fact, hunter-gatherers frequently lived well into their 60s, 70s, and 80s. But numerous individuals would die in childhood, or like women would die in childbirth quite frequently, or they would die from war or famine or something like that. So if we have a bunch of people dying at 0 and a bunch of people dying at essentially 18, because I mean like any guys in here, you know what it's like whenever you're 18, you just want to fight. And like anybody who's ever had a kid, it's just like, hey, coin flip if you're going to make it or not. If we add all of that together, or really sum and then divided by the amount of people that we're talking about, really people live a fairly long time. Everyone good with this? Right? So, yeah. Like next time someone says, oh, the paleo diet's stupid because they died at 40, punch them. Right? Especially if you're 18 and male and like you're really wanting to bring down our like lifespan total. Okay. Man, I can't believe I say or think some of the things I think. It doesn't matter. Here's a super cool thing that he talked about, and I'm sorry that you can't really see this. I'm going to tell you what all of this is. Physical inactivity up here. And inhibition of health-promoting proteins. So write that down if you don't know that. Here is Dr. Booth's idea, and like if any of y'all can't tell, like I know this guy and I think he's a genius. He's saying here, rather than like exercise causes changes to the body, he says, no, exercise doesn't cause changes to the body. Being physically inactive causes changes to the body. That's what's happening right here. So altering this around, physical inactivity inhibits health-promoting genes. So your state of existence is having health-promoting genes and silencing or methylating disease-promoting genes or proteins. Proteins, genes, I'm using those interchangeably at the moment. That's they're different, but here that's whatever. So does that make sense to everybody? That's really the main thing that I wanted you to get from this. Is that the whole theory from like our Paleolithic and earlier evolution is that physical activity is normal, and then physical inactivity is really kind of a problem of modernity or current now problems, right? So move around more. Something else here that I'm going to talk about. Here, I said this, and this, like, those of you that have like taken like biology classes, I love biology departments. It's it's one of my favorite things. Just one of the problems that I always had with it whenever I started reading all of this, they typically teach resting physiology, or like what happens in the cell at rest, right? Like what like those bio students, would y'all agree with that? Okay, like overall. And every time I would say, dude, this isn't normal. Like sitting down all the time, that is abnormal. We're not supposed to do that. We're supposed to like be outside doing push-ups, doing pull-ups in trees, and like chasing down wild boars and punching them in the head, right? That's what we're supposed to be doing. Picking berries or apple orchards, right? People do that up here, right? In Texas, we don't have apples, so it's we do, but like it's well, because that's where the Garden of Eden actually was, but whatever. So resting physiology is not normal physiology. There's an expectant amount of physical activity for the body to function. So here's an issue. Whenever whenever you're reading research, so if you take my research methods class, or if you, I don't know, go to Google Scholar and you just read things, here's typically what happens in these graphs. They compare two groups. Some type of good thing, meaning like reduction in body fat, like increase in VO2 max, increase in muscle strength, increase in whatever good thing you can possibly think of. They have a control group that is typically sedentary, doing nothing, right? Is everyone good with this? Like this is typically how it works. And then the other group is some type of special exercise. So CrossFit, eating an Atkins diet, like doing, I don't know, like Tabata training, which, does anybody do Tabata training? Tabata training? Does anybody know what Tabata means? Like high intensity interval training? Really? Nobody? Like, does anybody do group exercise classes, like Tabata classes? No? Man. All right, so well, y'all are the control group now, so y'all are the problem. So change that. Start doing Tabata. Read into it. It's like I had a whole story about like I actually met the guy like started Tabata high intensity interval training. He's the this is gonna be Tabata. All right, no stories. It's all right. I gave him a hug. But here, whenever we compare, like, say, all right, 5 sets of 5 on a squat, or CrossFit, or running however many miles, 10 miles a week, 20 miles a week, whatever, we're comparing that to nothing. So no shit, it gets better. No shit, your physical body gets better by doing something rather than nothing. So this is something that Dr. Booth always wanted. He said, this is what we actually should do, is have a sedentary control group and then a normal physiological control group. So here's the thing. Let's think about percent body fat. So most people want to lower percent body fat. Would y'all agree? Right? Okay. That's, I mean, like I totally do because, well, I'm single and I'm trying to change that. But, man, quit saying stuff. Whatever. So if we compared, like doing CrossFit athletes are super ripped, right? Does everyone agree with this? Right? Because they do this nonsense in like Madison, Wisconsin, which is apparently a place that I would never go to. But if we compared the special exercise doing CrossFit to walking 9 miles every day and fighting mammoths, what do you think would make a person leaner? I mean, like maybe they would have the same effect, you know, like whatever Matt Frazier is doing or Rich Froning Jr. or what, well, like they actually work out like quite a bit, like I don't know, like 70 billion thrusters or whatever, but that's, do you know how interpreting things can change when you just alter your mindset just a little bit, right? That's I don't know. I think this is cool, and if you don't think this is cool, memorize it because it's going to be on the test. So, all right. Here, just a couple of points. And I actually messed up on point number 2, so pay attention to this. Exercise is integral to human function. Right, so we've said that a bunch, obviously. Like, gosh, exercise is good for you. Everyone's cool with that, right? Right. Now, like, I will show you some like non-responder data from like Claude Bouchard, but like that's such a small amount of people, it probably doesn't even matter. This next thing. Exercise turns on good genes and suppresses bad genes. Cross that out. I messed up right here because I have this idea in my head that exercise is something that you do. No, exercise is the norm. Sedentary behavior, like, maintains good genes being on and suppresses bad genes. Oh, wait, hold on. Like I said that totally backwards, huh? Right? Thank you, Quinn. Thank you. It's I need to sleep more. So, like, being sedentary turns off good genes and turns on bad genes. So this is a super hard thing to keep in your mind. I mean, crap, I have a PhD in this, and I mess this up. Right? So you be better than me. Which most of you are taller than me, so it wouldn't be hard. Yeah. Being tall actually is pretty good. Like, you should look at the torque equation. Or the correlation between height and like IQ. That's pretty awesome. Okay, good. So understanding health, exercise, and disease from an evolutionary viewpoint is paramount for treatment and cures. So current science is somewhat limited right there. That's good. Now here's like a quote from the Frank Booth paper that I think is really cool. So back in the day, whenever people were like looking at hearts or whatever, they would have, instead of like comparing athletes to non-athletes, it would be like rich people versus laborers, people that worked in like coal mines, or people that worked out in fields, all that type of stuff. So the controls used to be physically active. You get that, right? Like if our control group was a laborer, or like say a lumberjack who's chopping wood all day, their heart's probably going to be bigger, right? And then we would probably start thinking like whatever your couch to 5K training isn't going to cut it, right? At least in comparison. Good, so hopefully all of that makes sense. Now genetics. A couple of memorizable stuff in here. And I'm going to point you to the parts that I really think are important. So really, there are numerous signals, but overall, there's one signal that I'm going to want you to know for endurance, and one signal I'm going to want you to know for muscle strength or hypertrophy. And hopefully, after going through all of this, you won't ask the question, is 3 sets of 5 better than 5 sets of 5? Or should I do 3 sets of 10? Like, should I run this amount of mileage at this % of my VO2 max? Or whatever. Because ultimately, on a genetic level, we're just turning on the light switch on one of these or the other one. Does that make sense? Right? So there's numerous like debates of like what's the best type of training. Well, you don't understand genetics if you ever ask that. You really just have to like turn on mTOR for resistance training or like RPS6. And for endurance training, you really just have to turn on something called PGC1 alpha. And once we do that, then we're off and running. Then it's just like magnitude or degree of turning that on. And like repetitive bouts of it. But here, let's get into this just a little bit. The central dogma theory, that's what it says up top. Central dogma theory. Now, this is a super old concept. I just want you to know it. It's archaic, essentially, and not really how it is. But the central dogma theory is the basic framework of genetic information flow. So know this. DNA. Transcription to RNA, translation to protein. Is everyone good with this? Good? So I'm going to use a couple of terms that like I'm talking about specific places within this. So DNA and transcription to RNA is typically called genetic expression. Now, all of it all the way to protein is called protein expression. So most things within our cell that are functional are proteins. That's why they're so important. Proteos, from Greek, actually means like the most important or of utmost importance or something. I don't understand Greek. Right? Okay. So here, that's all I want you to know. Understand DNA, transcription, RNA, translation, protein. Now, if you go beyond this, I want you to know the unidirectionality of these arrows is nonsense. It doesn't go one way. It actually goes back. Does anybody know what like a retrovirus is? So retro meaning backwards. So it like goes in through RNA, goes through the transcription, then does something stupid to DNA. Right? I'm not going to talk about that because, well, I'm not paid to talk about that. But overall, all of these are interacting with each other. We're going to talk about stuff called transcription factors here soon, where that's effectively a protein that's turning on DNA in order to induce transcription. But we'll get there whenever we get there. Okay, only a few parts of the cell that I want you to know. I don't really care if you know about the smooth endoplasmic reticulum or like lysosomes or the Golgi apparatus or anything like that. There's just 3 things, like the nucleus, ribosome, and mitochondria. Now, whenever we get into muscle, the sarcoplasmic reticulum, I will want you to know, but we're not there yet. So here, nucleus. This is essentially all I want you to know. It contains, man, one of my friends, Matt Romero, like whenever we were taking class, he would his birthday is on Valentine's Day, actually. So that's coming up. I'm going to call him. Okay, so each cell contains the human genome, usually one per cell. Now, in muscles, they're multinucleated. Know that. We have numerous nuclei in muscle cells. And that's going to be important for like muscle hypertrophy later on. There's something called the mononuclear domain theory. So nuclear means nucleus, right? And I don't care if you know anything about that yet, but I will bring it up eventually. Next, ribosome. Ribosome. Know this. Ribosomes are the site of protein synthesis or the site of translation. Ribosomes, that's where translation happens. So overall, with the central dogma theory, DNA is in the nucleus. Where does transcription happen? In your heart? I don't know. Yes, in the nucleus. Yes, Ari. Yes. Transcription happens in the nucleus. RNA, it's exported out of the nucleus, goes into the cytosol, or sarcoplasm, whatever you want to call it. And protein synthesis, or translation, happens in the cytosol at the ribosome. And technically, there's two different aspects of the ribosome. There's this 40S and 60S subunit. The only reason I'm bringing that up is if you ever want to read deeper into this. There's another term that I want you to know, and I'm not going to test you on it like anytime soon, but ribosomal biogenesis. So that is the creation of new ribosomes. That is one of the adaptations of lifting weights or resistance training or something difficult. Because it would make sense, right? The place where protein synthesis is happening, and if we want more muscle mass to be more jacked up, yoked, if we have a higher, what's called translational capacity, or more ribosomes, more places to do protein synthesis, we can be better at it, right? That makes sense? Yeah? Cool. I promise it seems like I'm talking like all this busy stuff, but whenever we come up to the test, I'm going to give you a test review, and I'll let you know exactly what I want you to know. Good? So does it sound like I'm talking in like a different language to anybody in here? No? Not to Hannah? That's because you're awesome, right? Best signature in the game. Yeah.
You're saying doing the like training, you're creating more ribosomes?
Yes.
Synthesize protein better for the muscles?
Yes. Yeah. Yeah. Yeah.
That's like an example of like a good gene turning on a good gene.
Yeah, that would be. Yeah, totally. Yeah, it's like I forget what it is, but like 2/3 of the RNA inside of a cell are ribosomal RNA. So that's one way how people like actually investigate ribosomal biogenesis. They just measure the amount of RNA. What is that? A nanodrop or something. Okay, moving on. Mitochondria, right here. Main thing with this, fuel oxidation, and in particular aerobic, meaning with oxygen, fuel oxidation. Another cool thing about this is it has its own genome. So your normal nucleus has about 20,000 to 25,000 genes. I don't remember which one it is. It's somewhere in there. Probably need to ask Watson and Crick about that. Not I. Now the mitochondria has 37 genes. The only reason I'm saying that is it might be a bonus question at some point. 37 genes, and it's my favorite number. That's why I remember it. Now, here's kind of a cool question of like heritability stuff. Which parent do you get your mitochondria from? Your mom, right. Because like a sperm cell doesn't have mitochondria in it. Is everyone good with that? So if if you had to construct a human being, not with like CRISPR, Cas9, like whatever stuff, but like you just had to like pick two humans and make them breed like the old-fashioned way to make the best aerobic athlete you possibly could, who would you rather have a super high aerobic capacity or good at endurance sports, the mother or the father? Mother, right? So yeah, so since we're getting all of that from there, and like my mom has a super low VO2 max. Now I don't know if that's because she smokes all the time or if she was just born that way, but coincidentally I have a pretty low VO2 max too, so that's kind of cool. All right, moving on from there. Okay, so the 4 bases. I do just want you to know these, the purines and pyrimidines. I doubt I'm saying pyrimidines right. That's hard for me. But adenine, guanine, cytosine, and thymine. These are just the general like nucleotides that are going on in your genetics. Now, A's go to T's, G's go to C's, right? I could ask you a question about this. However, there's a lot of other stuff I'd rather ask you questions about. Now, one thing that I'll probably do if I'm ever talking about this, guanine, I've almost always called it guanosine in my head because it with cytosine. So if I make that mistake, I mean guanine, so my bad. Those going together. So a couple of definitions. A nucleotide, I would know these, right? A base along with carbon, sugar, and it's like a pentose, so 5 carbon, sugar, and a phosphate, PO4, 3 minus, something like that, -3 charge, I believe. So that's a nucleotide. A gene is just a sequence of nucleotides. So they can range in just a couple of nucleotides to a bunch of nucleotides. Next thing, amino acid, the building blocks of life. That is 3 nucleotides. They have different charges and different structures and things. I want you to write something in here that I don't have up here. Also called a codon, C-O-D-O-N. 3 nucleotides is also called a codon. We could talk about anticodons, but I'm not gonna. So after that, a sequence of numerous amino acids, which is 3 nucleotides or a codon, is a protein. So that's effectively just a bunch of amino acids. At least that's how I remember it. And there's a bunch of stuff we could talk about, like alpha helices, beta pleated sheets, tertiary and quaternary structure. I'm not going to ask you about that because I don't think memorizing that is really going to help you. But with the different charges of the amino acids, that can alter protein structure and therefore function. So most of the time, there's kind of these globule shapes of proteins, and they will have a part inside of them called a catalytic pocket, meaning that's kind of the functional part of it. And if we change one of the amino acids, then we can close that or open it in some way, rendering it positive or negative. So whenever Quinn was talking about turning on a good gene, this could be a potentially good mutation. So if there's a particular protein that's related to exercise, if we alter one of the amino acids in it, and we'll get there, I think it's on the next slide. If we alter one of the amino acids in it, that makes it better at transporting glucose into a cell, into a muscle cell in particular, that might confer an advantage in terms of whatever sports ball thing that you're doing, right? Patrick Mahomes or whatever, he has good genes for throwing balls, I think, right? Maybe? No? You think so? Okay, yeah, that's, gosh. I think he looks like Patrick Star from SpongeBob, but that's just my opinion. It's just my opinion. Good luck, good luck. All right, next thing. Something I want you to know. It's called a SNP, S-N-P. I want you to know what this stands for. It stands for single nucleotide polymorphism. So single nucleotide polymorphism, essentially all that is the alteration or change of one nucleotide within a codon or amino acid. So it looks something like this. So say we have this genetic sequence that, down the road is going to end up being a particular protein. If we have some level of mutation to where we go from like an A to a G or to a T or whatever, just whatever alteration, that can alter what I've been talking about, the protein function going on. So here's something that, I don't know. Let's take an enzyme, for example. So enzymes have like a catalytic pocket that has some type of interaction with some type of substance. And like, I don't know, like I'd think about it kind of like this. Like we have to, once glucose gets into a cell, we have to do a bunch of stuff to it before we can get energy out of it. And this might kill somebody, but like let's just think about it like this. Say we have like a particular enzyme that has a normal interaction with glucose, so this normal shape, normal function. If we have a single nucleotide polymorphism that's a big deal, we might alter the shape and therefore not have the typical like reaction that we would with glucose. So then, say, sprinting would be hard. Does that make sense to anybody? Like that's somewhat related to something called like McArdle's disease, where you actually can't access like the glycogen in your muscles. But we probably won't really talk about that too much. Here is a typical SNP that most of you have likely heard of. Who's heard of myostatin? Hannah has, okay. Anybody else? Myostatin? A couple of people? Okay. So this is essentially just like a mutation that's going on, right? So myostatin, it's produced by muscle to inhibit myogenesis, or, like growth or production of like new muscle and all that type of stuff. What it really is, it's an 11 nucleotide deletion in the third exon. That doesn't really matter so much. Effectively, what this means is a frame shift. So one of the nucleotides, whenever we were doing transcription, got deleted for some reason. So whenever that gets deleted, the reading of the rest of the protein makes it to where this myostatin is non-functional. And that one little deletion in nucleotide makes this happen. Belgian Blue Cow, has anyone ever heard of this? Right? The Arnold Schwarzenegger of cows, right? Yeah, which, I mean, Arnold Schwarzenegger didn't have like a myostatin deficiency. He did Dianabol, so it's different. This also happens in bully Whippets. So that's a dog. That's really cool. So right here, this Whippet doesn't have the myostatin like augmentation. However, this one does. Do y'all see how much more jacked it is? Right? This is the alteration of one nucleotide, meaning like that one nucleotide goes away. Is that crazy to anybody? Right? I mean, and like whenever we're thinking about like transcription, translation, a bunch of nonsense is going on all the time. So those things, it might be a little bit more common than we think. So effectively, I just wanted to show you this picture to like try to kind of make some sense of this. Say like there's a normal reading of like, hey, like the AUG, that's methionine, AAG, lysine, so on and so forth. Here, if we just take out one of the U's, one of the uracils, that's RNA, like I didn't tell you to know that, then that completely alters the rest of the reading of the protein. So that's what a frame shift is. Is everyone good with this? That makes sense? Right? So you could explain to me on test what a frame shift is? Metal. Good. All right, there's like a couple more that I want you to know. And I'll talk about it just a little bit. So a silent mutation, I want you to know what this is. Silent mutation is the interchanging of one codon that leads to an amino acid that has effectively the same function as another amino acid. That would typically be in that spot. One more time, Sydney, is that what you said? Okay, gotcha. A silent mutation, meaning that, like just gonna backtrack just a little bit, that there is a mutation in the genetic code, yet you don't see anything. There's no phenotypic or outwardly presenting differences. So what that is, a change in one codon, or like one nucleotide within the codon, from one amino acid to another amino acid that has essentially the same function. The next one, my favorite one, because it's funny to say, a nonsense mutation, right? I have a meeting today at noon, and frequently in meetings, I yell out nonsense or tomfoolery or something like that. And everyone in the meeting, they don't know that I'm talking about a premature stop codon. Meaning, whenever I say nonsense, that the meeting should stop now. Right? Anybody? Okay. That's, gosh. Okay, so a nonsense mutation is a premature stop codon. So we're reading to make a protein, and then this nonsense mutation, UAG typically, or UAA, or something like that, says stop making the protein. Meaning the ribosome is like, okay, done, right? Not continuing the protein in any way. So premature stoppage codon. I had a whole thing I was going to talk about with alpha-actinin and 3 here, but I'm not going to right now. The next one is a missense mutation, which is just, it's fairly similar to a silent mutation. However, and like it depends on where you read the definition, but at least how it was taught to me, was it goes from one amino acid that has a particular structure to another amino acid that has a different structure. So like, let's look at these, arginine and threonine. Where are they? Where are they? Okay, so down here, threonine, that's polar, right there, polar, whereas arginine is positive. So it would be like essentially you swap out one of these amino acids for one of those, or one of those for one of those, or one of those or one of those. Now, I'm never going to have you memorize this. Just that's overall what I want you to know about a missense mutation. All good with that? Yeah. I mean, ultimately, it'll change the shape of the entire protein, right? Now, like a silent mutation would be like looking up here, going from glycine to alanine, because it's the same. They're both nonpolar, and if we interchange those, it doesn't really matter so much, right? Now, it's interesting, but it's not super meaningful. Okay. So next time, we'll get into how gene signaling works. Make sure to watch the RFD stuff. Be ready for that. And don't eat before lab. Cool? Because if you throw up, that's on you.